Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning, everyone. Today we will explore brain plasticity and how neural circuits adapt with experience and injury. Brain plasticity refers to the capacity of the brain to change its structure and function over time in response to internal and external influences. I want to begin by distinguishing two broad forms of plasticity. Functional plasticity involves changes in the strength of existing synapses, while structural plasticity involves growth or retraction of dendritic spines, axonal sprouting, and even changes in regional volume. Both are central to learning and recovery. At the synaptic level, we see mechanisms such as long-term potentiation and long-term depression. LTP is a persistent increase in synaptic strength following repeated coincident activity and supports memory. Encoding limited weakens synapses and helps refine circuits. These processes reflect Hebbian principles, often summarized as cells that fire together, wire together. More precisely, timing matters. Spike timing dependent plasticity shows that if a presynaptic spike precedes a postsynaptic spike within a narrow window, synapses are potentiated. If the order is reversed, they are depressed. This millisecond precision affects network dynamics and learning rules in cortex and hippocampus. Structural plasticity operates on longer timescales. Repeated practice can enlarge dendritic arborization and increase spine density. Neuroimaging has revealed structural correlates of expertise. For example, studies of licensed taxi drivers in London found increased posterior hippocampal volume linked to extensive spatial navigation experience. Animal experiments illustrate causality. In enriched environment paradigms, rats raised with toys and complex layouts develop greater synaptic density and higher levels of brain-derived neurotrophic factor, or BDNF. BDNF promotes synaptic growth and is critical for many forms of learning-related plasticity. Plasticity is not confined to development. Although critical periods exist for some circuits like the visual system, where ocular dominance columns are shaped during early life, adult brains retain capacity for remodeling. Adult neurogenesis in the dentate gyrus of the hippocampus adds new neurons that can integrate into existing circuits and support certain types of learning. Clinical examples highlight applied significance. Consider stroke recovery. When a cortical region is damaged, neighboring areas can undergo cortical remapping and take over lost functions. Therapies that harness this capacity can improve outcomes. Constraint-induced movement therapy forces use of an affected limb and promotes functional reorganization. Phantom limb phenomena also reveal plasticity. After amputation, the representation of the missing limb in somatosensory cortex can be invaded by adjacent body part representations. This reorganization underlies altered sensations and sometimes pain. We should also consider timescales and reversibility. Synaptic modifications can occur within milliseconds to minutes. Structural changes emerge over days to months and can be partially reversible depending on ongoing experience and neuromodulatory state. Experimental manipulations that combine behavioral training with targeted neuromodulation or pharmacology show promise. For instance, pairing training with non-invasive brain stimulation can boost LTP-like processes and enhance rehabilitation gains. These interventions aim to amplify naturally occurring plasticity. To summarize, brain plasticity encompasses multiple mechanisms, from spike timing, dependent synaptic changes, to large-scale cortical remapping. It is experience-dependent, and it underlies learning expertise and recovery. Following neural injury, plasticity can be adaptive or maladaptive, depending on context. For instance, when stroke patients undergo constraint-induced movement therapy, they often show cortical remapping that corresponds with functional gains. In closing, I want you to think about plasticity as a spectrum of processes that can be measured electrophysiologically with LTP and limited paradigms imaged with MRI and manipulated with behavioral and pharmacological tools. Understanding these mechanisms will advance both basic science and clinical practice.
Listen again to part of the lecture. Then, answer the question.
For instance, when stroke patients undergo constraint-induced movement therapy, they often show cortical remapping that corresponds with functional gains.
This concludes the listening section. For more TOEFL practice tests and study materials, visit Spinta.org.